presents Dr. Tom Kipps. Just quickly, by a show of hands, how many of you have seen Dr. Kipps in consultation? Yeah, all right. And if we added Dr. Castro and Dr. Choi, it would probably be almost the whole room. So Dr. Kipps is the professor of medicine, the Evelyn and Edwin Task Chair in Cancer Research, Deputy Director of Research Operations at UC San Diego Moore Cancer Center, where we are now. He has a PhD in immunology and an MD from Harvard, so he's one of those brainy MD, PhD types that are really moving the bar forward. He did his residency in internal medicine, hematology, and genetics at Stanford. He has a reputation around the world for his research in gene therapy, immunology, and other fields. He's standardized protocols for the treatment of our disease. He's offered more than 300 publications, and as we all know, he's the lead investigator on a number of important researchers, but he's just, as anybody who's ever seen him or knows him, he's just a wonderful person in terms of taking care of his patients, caring about his patients. So it's a real blessing to introduce Dr. Tom Kipps.
Thank you very much. It's a pleasure to be here, and actually it's a pleasure to see many familiar faces and friends, and I really appreciate everyone coming. After that introduction, I think the cue I should take is just to sit down. I'm done. But I understand that a number of people are still caught on the freeway. One thing that Southern California is not in short supply of is traffic. But I do think I would like to get started just on the interest of keeping within the schedule, at least as best we can. I think that we have my list. So I just would like to say if okay, we're getting set up here. Anyway, I was asked to try and provide an overview, and I first want to say that Dr. Lipman, who is our Cancer Center Director, wanted to actually be here, but due to a family emergency, I think he was unable to attend, and so he sends his regrets. But I do want to welcome you to San Diego, and many of those who have been here before know this place, but I think it's important not just the place, but the types of activities that are going on here and elsewhere to try and understand more about this disease and how to better treat patients with this disease. And so I would like to introduce the concept of the CLL Research Consortium. As you know, this is an organization that has been around for well over a decade, and it's been meeting on a semi-annual basis, and our annual meeting is coming up. And we wanted to just give you an outtake of what the CRC actually is, the CLL Research Consortium, and also provide some overview of the progress that we have in CLL to give just a touch of some of the biology and how that's leading into the new treatments that are currently being offered for patients. I think that it's fair to say that the CRC is a consortium of investigators, not just here, but around the country and even in parts of the way in England. And you can say that it has early beginnings in the past millennium. So it's hard to recognize these people, noted CLL investigators, but perhaps as in this picture, which was during our early meetings with Conti Rai here, and Dennis Carson, and John Reed, and Bill Plunkett. I think there's John Driven, and Carlo Croce standing there, and you see Michael Keating and Michael Grieber. There's a young John Bird. There's also Laura Rosenti and Barsha Gandhi, and a number of others, Lee Nadler. So this was a group that early on decided that we should really try to pull our collective resources and band together to form a consortium. There are many different attributes that were borne in mind, is that some people were doing work in genetics, some people were doing work in immunology, some people were doing work in biochemistry. It was thought that by coming together, we can complement each other and try to move the field faster along. And I think that that's happened, and this is our current status with the consortium. And some of the noted people, some of which can't be here, but some are here. Let's think about Bill Weirda, who's leading the leukemia service now for CLL at the MD Anderson. Carlo Croce also could not be here, but Jeffrey Jones, who is one of the leading clinical investigators now at Ohio State, is here. And John Bird and Amy Johnson will be here shortly. We have Conti Rai, who also was hoping to attend, but he sends his best, along with Neil Kay, who is here, who's going to be speaking about some of the work that they've done with the consortium with the Mayo Clinic. And we have John Gribbin, as I mentioned here, on this faraway island off of the northwest. So I think that what is a consortium? A consortium is a group of people trying to get together to do more than what they can do by each of them alone. And I think it has been really a pleasure to be able to interact with this group. They've been very dedicated and trying to apply their skills in various ways and complement each other. And this program actually has an ultimate goal. It was quite clearly stated in the application we had. Really, the goal is to try and find better treatment strategies for patients with CLL. Because I think in the long and short of it, as much biology and genetics as we're doing, it's really important to set the goals straight so that we can make this have resonance in what we hope to achieve in the long term. And I think that there's other collateral benefits, of course, but finding more treatments for CLL will help our understanding of how to treat lymphoma in general, and also to have novel therapies for patients with solid tumor malignancies. And there could also be an assist in the evaluation of these targeted therapies, how we can better tweak them or put them in combination. And I think that it's been clear that to really understand and be able to effectively treat patients with cancer, we need to understand more about the disease of cancer. And too much in biology, we focused on cell lines or model systems, which are great, but it's not the real thing. And so oftentimes we're forced to look at shadows, but the ability to actually look at the tumor cells and say, well, this is what it is, take it apart like you would the kitchen clock on your bedroom night table, and be able to figure out how it works and then try to intervene, prevent it from getting worse. Now, this is how the consortium is organized. Studies in genetics and biochemistry, immunology, and pharmacology, different programs and different cores. Carlo Croce led the genetics effort. He's a really fabulous guy. He's originally from Rome, and I must say, I never would like to bet against this guy because of tremendous intuition, was able to make some very important discoveries in the genetics for not only CLL, but other cancers and lymphomas. And actually, through the work of the consortium, had helped to find one of the key drivers, the mutations that might be affecting how patients may develop this disease, and it's been very helpful in terms of understanding how we can try to intervene. We have Maurizio Collecchia and Varsha Gandhi leading Project 2. This was formerly led by John Reed, who now heads Roche. It's a big job for him, but they tapped him to lead the entire Roche research efforts. Maurizio and Varsha are still carrying on the efforts to understand why leukemic cells are so hard to kill inside the body. And there's a protective mechanism for keeping the cells alive, which we all call BCL2. And that's brought about the targeted therapy against BCL2 to try and break away that shield so that we can actually have the cells be able to overcome this resistance to apoptosis. I lead a group in immunotherapy, which is looking at different immunomodulatory drugs and different immune therapies, and assisted by Dr. Castro and Dr. Choi in the clinical investigative parts of that. And that's been a pleasure to work with these two colleagues. The area of immune tolerance and transplantation is Dr. John Gribbin, who also has degrees in immunology. He's done some really fantastic contributions to understanding what is the basis for some of the immune suppression that we see. I mean, one of the major facts of patients is that they have problems with a suppressed immune system. It just doesn't want to respond against infections. And so he's been working hard to find out how the T cells, which are part of the immune system, how they are turned off by exposure to the leukemia cells. And that really resulted in some very important insights, and it led the way to thinking about how to use such things as called immune checkpoint inhibitors, which are ways to take off the brakes of the immune system so you can get the immune system to act against the tumor. And this is something that has been very helpful in solid tumors now, and it may be applied in chronic lymphocytic leukemia. We also have Bill Plunkett leading pharmacologic efforts. He was actually the one that helped devise such combination therapies, purine analogs, fludarabine, also some novel therapies that are currently being done for patients with certain genetic lesions. And so these are helpful because as we find more about the genetics of this leukemia, we might be able then to go after them in a more focused way. And then we have the Project 6, formerly with Dr. Michael Griever, but now with Amy Johnston and John Bird, many of you know. And we're fortunate to have also Jeffrey Jones here, who's really, I think, a very important investigator at Ohio State, and he'll be talking today about the kinase inhibitors. And we have other clinical investigators too. Dr. Neil Kay, he was actually here in San Diego for a brief while some years back, but has actually been for the last many years in Mayo Clinic, and I think really has helped shape the Mayo Clinic's investigation of chronic lymphocytic leukemia, bringing in young people such as Tate Schoenfeld and others. And I think he's had a long-standing interest in this disease. And it's important to note that people go to different institutions, and it's the people that drive the agenda. And I think Mayo Clinic would be doing other things if it were not for Dr. Kay in terms of looking at CLL. And we have other clinical investigators who can't, unfortunately, be here. Dr. Ryan mentioned, Dr. Jennifer Brown at the Dana-Farber, and Dr. Jackie Barrientos, who's going to be here shortly, but not today. And then we have a high school picture of Andrew Brees. He hasn't changed, but he helps with the administrative core, bringing this together, keeping folks playing well in the sandbox, and also bringing together people like this meeting here. So appreciate his efforts. Donna Newberg, I believe, will be here as well. She's the statistician. What I really enjoy about Donna is that, you think about statistics, it sounds like pretty wonky stuff. I've always wondered, though, what this, is this one of those invertible 8 balls that you ask the question? I call her on the phone when I have these intriguing questions, and she always seems to have an answer for me. But no, she's a wonderful statistician because she wants to understand more about the disease. She truly cares, and so it's nice to talk to a real person who can put the statistical analysis into frameworks that we can all understand. And of course, we have Laura Rosenti here. I don't know what she's getting out of the freezer banks, but I'm sure it's important. And she has been managing our tissue core now for since the inception of this, and having to keep track of patient samples is no small feat. We have over 6,000 patient samples, many serial samples, and she has every one of them categorized and coded so that we don't breach patient confidentiality, but she's able to pinpoint that cell that was taken from that patient on that particular day and having clinical data that can match up to it. So it's all done in a fashion. It's really been a tremendous thing because if you think about it, we don't know how the future is going to be with any type of therapy. And so by being able to collect the cells from patients and being able to bank them down, we can actually then know what the outcomes are and then go back and see, are there clues that could help predict those outcomes? And that's so critical because you can move the field much more quickly if you can go back and do those types of experiments in real time rather than having to start all over again with another group of patients taking the same drug. So that's so important. And we have Bill Wirdy here, who is also here formerly at UCSD, but now I mentioned leads the CLL service. Dr. Keating is still present, but has taken Bill Wirdy's taken his place in guiding that effort at the MD Anderson, and he's a terrific guy. He's here to talk about some of the newer therapies that are being introduced at the MD Anderson with the Consortium. So I think as a model system, CLL has many different advantages, I think, but it's more important to focus on what can we learn from this, process. Having access to cells is really kind of peculiar because if you think about it, how many cancers are there where you can actually just get the tumor cells out by taking a blood sample? And I think part of our problem in cancer research is the inability to do that. And so we can't really understand what we're dealing with, and we can't understand how it responds to our treatment. So if a patient gets a drug, you do an x-ray, and you see things shrinking, but you don't know really what's going on. So we have the opportunity to look at that in chronic lymphocytic leukemia. And so we can also look at how things may change over time. And I think that we've come to understand that there is somewhat of an evolutionary process. We're trying to understand that to stay on top of it, because there could be an event that leads to development of the leukemia. Now with next generation sequencing, we're trying to understand how this process may go, where you may have other subclones taking off that have different characteristics. And we need to know this so we can come prepared not to just take care of the cells in white, but also take care of the cells in red. And so that these may present themselves after treatment to be the predominant population. So things can change, and we need to understand that change so we can be able to target our therapies at this or be able to take measures to prevent that change. That's a very important question that we have. So I think the other aspect is the biology of this disease. As you know, the leukemia is a B cell, which is a cell of the immune system, so much attention has been focused on the biology of B cells in general. And all these things that you see here listed are all the proteins that B cells have that are also present on the leukemia cell. And I've listed here in red, these are two proteins that are targeted by monoclonal antibodies that can bind to the leukemia cell and then have very strong activity against the leukemia cell. Now, one of the things we're having to face is that these antibodies will bind to normal B cells too, and in the case of this particular protein, CD52, it's also found on T cells. This is a protein that's targeted by an antibody called Campath or alatuzumab. So I think that what we have is these markers on B cells, and we can actually direct therapy against B cells, and that's been very helpful in defining therapies that have improved patient survival. But we still have to be mindful of the fact that we still would like to have B cells around, and actually if we were able to direct therapy against the B cells, we hope that they would recover after successful therapy. I've been asked to present this slide again by popular request, but so somebody asked for this again. But actually, if you understand the mechanics, it sounds very complicated, but you know, I've talked to New Yorkers and they have all the subway systems to heart, and they know every street and cranny in Brooklyn, and they can get around any block. And so what happens is that understanding these pathways under the surface of the cell allows you to understand routes which can block cell death. For example, it's the yellow here. And if you can then throw a monkey wrench into that pathway, you can block that pathway. And the fortunate thing is that many cancers are somewhat like schoolyard bullies, and if you take away what's driving them, they sort of can become wimps, and they're less resilient than normal cells. And as a consequence, we can maybe intervene with some of these pathways and achieve these blocks. And I think that's been very helpful in defining new types of therapies. Now, this is the leukemia cell, of course, and so I told you there's a lot known about the biology of these proteins and how we can target them with monoclonal antibodies. But we now know it takes many cells. It's actually the environment. And there's kind of a process that goes on, is that the leukemia cell will conscript other cells in the body to help it survive or help it to grow. And we know that this leukemia cell is very fastidious, which means it really can't survive on its own. It requires help from its friends. And understanding these friends can help us to define ways that they communicate back and forth. And that provides then clues as to how we can intervene with either the communication or these other accessory cells to define therapies that really are helping to treat the leukemia. So I guess some have said it takes a village. Well, here it takes a microenvironment. So a lot of attention has been focused on this protein here, which is an antibody, because this is a B cell. They make antibodies. And there's only one type of antibody on the surface, and that serves as a very important antenna to receive signals from the outside environment. So what B cells do is typically have an antibody, and if they bind to a protein, they can become stimulated, and they can grow, and that's how we develop immune responses against infections, against viruses and proteins. We have B cells that each make antibodies, and it's sort of like, select me, and the antibody that's made by the B cell is only one type. And if that B cell is fortunate enough to win the lottery and bind to the target protein antigen, then it could be expanded to develop the immune response. And it's no different for this leukemia cell too, in that they have this antibody, and it serves as a very important receptor on the surface of the leukemia. And other facets of this disease have come out, namely the signaling pathways. You know, we've heard about ZAP70, for example. What that does is it seems to make the signaling of this antibody on the surface more effective, and so you get this being stimulated by some protein in the outside environment, or maybe even a cell protein, and you can get an amplification of the signal. And what currently, one of the current models is that those B cells, or those leukemia B cells that have more effective receptors, may tend to give more growth stimulation to the leukemia cell, and that might account for why the leukemia cells may be a little bit in overdrive compared to patients who do not have as much of a functional receptor. That's an attractive model because it indicates that if we attack the signaling pathways for the antibody, that might be effective in treating the leukemia. And so that's actually been done. So here's the antenna that I talked to you about. This is the antenna on the surface of the B cell, and it does have these signaling pathways. You can map this out like the subway maps of New York City. And you can see these different proteins, they all lead then to signaling and down to proliferation, survival, and cell migration. And so these different pathways are here, and they've all been mapped out. And so some of these proteins are enzymes that actually work to modify their target proteins. And those enzymes can be inhibited by enzyme inhibitors or kinase inhibitors. And so there have been a number of them that have been developed. This inhibits this enzyme called SYK here. Another group of inhibitors against this protein, BTK, which is involved in this process. And then, of course, we have idelalisib, which is another inhibitor of this complex here, PI3 kinase, and there's a number of other ones down the pike. And so these inhibitors allow us to throw monkey wrenches into the signaling pathways. And because the B cell needs this sort of tonic signaling, it's sort of like having your antenna taken down and you can't see your favorite show. So having your cable cut, I guess that would be more current. And so this does affect the B cell, but there's also another aspect to this that is very important to take notice of, is that this is the leukemia cell again. It has another important receptor here. This is something that was defined now over a decade ago with Jan Berger, who was formerly my lab, but this is an important receptor. It acts like a nose to the cell, which allows the cell to recognize there's a chemical out there that it has to go towards, so it allows the leukemia cell to migrate actively. You hear about the pheromones of the moths that can attract another moth 25 miles away? Well, this is true for the leukemia cell. They can attract by this process to get to those sites where they can get up with their buddies and be able to have this microenvironment that I talked about. So this allows for this recirculation. We know that this is a very dynamic process now, as the cells will go from, here's a lymph node cartoon, it goes from the lymph node and goes into the blood vessel, and then they eventually come back and go into the lymph node again. So there's a very dynamic process of going back and forth. And it's somewhat like, this is maybe considered like your home, and this is like the freeway, I-5, and you have to go to work and what have you, and then you go back to home and you get your rest and survival benefits. And so maybe keeping the cells outside of their home microenvironment may be a bad thing for them. And that's actually what I think is the major mechanism of some of these kinase inhibitors is actually doing. It's actually blocking the homing mechanism more so than the receptor signaling of the Ig. So if we block that signaling process, the cells cannot go back into the lymph node, and they tend then to accumulate in the blood, and the lymph node then will start to shrink. And that's what we see with some of the more modern therapies with ibrutinib and idelalisib and other kinase inhibitors. And so this has been advanced. So the question still remains, why, if we can block these cells from getting back into the lymph node, why are they still hanging around? And what can we do to close the deal and get rid of them altogether? Can we also understand more about this microenvironment and how its signals convey survival signals, which may be sometimes given to cells at a distance? We know that now, that there's survival signals that may act at a longer range and may be affecting the cells within the circulation or beyond. And I think those are important questions for us to address if we're really going to close the deal and be able to take patients who are on some of these therapies and be able to get rid of the disease entirely. Now, this is a CLL research timeline and development. So you can see I've gone back here to the 1970s, and this is how we progressed in our knowledge of CLL, because not much was done, and we had the Rye and the Binet staging systems in the mid-70s. This is very important because what these staging systems actually discovered was the importance of the marrow function. So your bone marrow is very important. Without your bone marrow, we cannot make our red cells, we can't make our platelets, we'd be in a sorry strait. And what this leukemia does is it tends to home to the bone marrow by these same signals that I mentioned, and it interferes with the ability of our normal bone marrow to function well. So it sort of occupies some of those same niches that are required. These are the nurseries for making our red cells and our platelets and everything else. And so what they recognize is that by patients, if they have either anemia or low platelet counts, that's actually not a good thing, and you should try to do something about it to start therapy. That was the major thing with the staging systems. Then there was also the discovery that you have very restricted types of antibodies. That was done over 2 decades ago. Certain V genes are being used, and we know that you can stratify patients, this is in the late 90s, patients into high risk or low risk categories by virtue of whether their antibody genes have mutations. And we're still not exactly clear how that associates with aggressive disease, but some clues are the fact that we see other genetic changes like the 17p, and now more recently with next generation sequencing, we're seeing other types of mutations which may drive the disease, not start the disease, but help to drive it if they should occur. And then the data identified this other protein, ZAP70, and that gave us clues to the fact that the receptor signaling might contribute in some way to the evolution of the progression of this leukemia. And so that was a very important concept. Also with the microenvironment, chemokines, was done in the late 90s to identify these, and that resulted in studies which helped to define that the fact that this leukemia is really growing and dying at the same time. The heavy water studies, patients were taking heavy water, which is not radioactive, but could be incorporated into the cell. And when they stopped taking the heavy water, you can measure how much of the heavy water was still retained by the cell. And what we found is that some patients were making a lot of leukemic cells every day, and they were just dying. So it was a very dynamic process. We have to understand how we can intervene on that process. But that resulted in the use of these tyrosine kinase inhibitors, TKIs, such as ibrutinib and idelalisib. Then we have the knowledge that BCL2 is expressed at very high levels in CLL. This is like the shield that shields the enterprise from the attacks from the photon. So with chemotherapy, with BCL2, we're not able to kill the cells as easily. Carla Groschi, of course, identified these so-called small RNAs, which can regulate BCL2, and that is very important because it allowed for mouse models to be developed. And then we now have the BCL2 inhibitors. These are small molecules that can go in and target BCL2, and these are proving to be very effective against the leukemic cells because they require this BCL2 in order to stay alive, more so than normal cells. And then, of course, the gene therapy efforts. Actually, Bill Ritter, when he was here, led several gene therapy trials, finding also new modulatory drugs. And now we have the CAR T cells as another matter, and we have the ROR1. So these are some of the developments that we have. And I think that these are the drugs that we have. The first drug, as you know, is chlorambucil. That's been around for some time. We don't know exactly when. And we're not really sure when it was developed, but we're probably sure that it was on a Tuesday. But it's been around forever, and that's been the mainstay of therapy. And then also other drugs such as cyclophosphamide and glucocorticoids, and things like prednisone has activity, and sometimes at high doses can be very active against leukemia. And then as we progressed, we had these so-called purine analogs, such as fludarabine or pentostatin, and they were heralded as the new drug and different than chlorambucil. And yet, more recently, I guess, when you looked at the data, it's hard to know whether fludarabine has more activity than chlorambucil, although at the time it appeared to. I think you can see a difference, but you really have to have quite a few patients being treated with one type of therapy or the other to find that difference. Then we have the combination of drugs. This is purine analogs with an alkylating drug like cyclophosphamide. That's FC combination. This is another alkylating drug that was actually approved in October 2008, so it's a latecomer, but actually this drug has been around for over 30 years, which is late to come into the CLL world. Then we started seeing monoclonal antibodies. This is the CAMPAC, which is alemtuzumab, developed in the early 2000s. And this is a very effective drug, but it also has to be used in the right way because this antibody is very effective in clearing leukemic cells, but it seems less effective in clearing the cells if they're in the lymph nodes. So what we have is that this has to be used on patients perhaps that have very small lymph nodes, if at all, and that can be very effective in curing the leukemia. Then we have the advent of combinations with rituximab. rituximab has been used as single agent, but these combinations show that you can actually improve upon chemotherapy, heralded the era of chemoimmunotherapy, where you have antibodies and chemotherapy given together. And the study was one of the cooperative group studies showed that there really is a survival benefit if you compare patients who take chemotherapy alone versus chemotherapy and antibodies together. Patients taking the chemoimmunotherapy actually survive better than patients who took chemotherapy. And then we have the more recent antibodies, ofatumumab, which is another antibody against CD20, and then the combination trials. This was a German study that actually showed that if you added rituximab to this combination, you actually could improve the treatment outcome of patients as opposed to giving the chemotherapy alone. And then we have the advent of the new generation anti-CD20 obinutuzumab, and then we have ibrutinib approved in April, and then idelalisib was approved shortly thereafter in July. And so we have many more drugs now to use that we didn't have before. So if we look at the roadmap of drugs that are currently approved for treatment, we've always had these alkaline drugs for some time. Monoclonal antibodies are a newcomer, and the purine analogs we've had for a while now. But these new drugs, the kinase inhibitors and new monoclonal antibodies, actually have been approved just only recently. And it's gratifying that we have more tools to use against this disease because in part of fact, it really is important that we don't hammer away with the same tool each time. And I think part of the problem with chemotherapy, one of the major problems, is if that was the only hammer, then everything is a nail. And as a consequence, patients were given more chemotherapy than they could tolerate and developed resistance to chemotherapy. So the advent of these new therapies is really making a difference. Patients are living longer and healthier lives. It's really very gratifying to see, and we hope to be able to eventually make good on that promise to see if we could hopefully provide curative treatment strategies for patients with CLL. Other agents that are under study, which a host of these BCL2 antagonists, and this one may be approved sometime perhaps at a year's end or maybe next year, and some of the studies are looking very promising. This ABT-199 venetoclax, there's other inhibitors of BCL2. We have immune modulatory drugs, and there's a new one that's being developed. And it looks very promising. It's probably easier to take than the drug before it, lenalidomide. And we have novel monoclonal antibodies targeting other proteins on the leukemia cell and development antigens such as the ROR1 antibody and development of CAR T cells. And there's other kinase inhibitors. I think once you have a successful drug, it's sure that there's maybe 20 different companies that want to develop an also-ran drug. So we've got maybe 20 different BTK inhibitors and a host of anti-PI3 kinase inhibitors. So this is going to be challenging to sort it out, but I would say that if we can find out whether some drugs may be tolerated better by some patients than others, it would be helpful to have more options than to be limited. So it's always a good thing. So this is a meeting we had in 2008, and you have on your pamphlet there this person here, Miles Pendleton, who was really our patient advocate, but also worked on the congressional committees and really helped to convince Congress, and also we had to help convince the NCI, because one of the problems that we've had in the research on CLL has been a little bit of complacency, obviously, because we have other types of cancers which patients may die within 3 to 6 months. It is a disaster, but the problem is getting at to try and figure out how to treat those patients has been very difficult because of not having access to the tissue, not having any models to test, not having any new drugs that you can develop. So we end up trying to address these needs, and I think it's very important, though, that in the treatment of CLL, convincing that this is a very worthwhile and important goal to have in mind. And the important point that I always make is that if we learn how, by studying a cancer really well, to try and dissect it so that we can cure it, I firmly believe that we'll not only crack open CLL and be able to provide curative strategies for CLL, but other cancers as well. Because there's not that much difference. Cancer is like us. It's something that's derived from us, and there are programs that the cancer may have that really could be understood, and then we can intervene with those programs to provide more effective treatment. But this is a way of saying thank you to Miles, who unfortunately succumbed to his disease, and he was a great advocate for us. And it's just also to say these people are the members of the CRC at the meeting. They're pointing to him because we were actually saying that this is really all about you. It's all about the patients. And I have to say this is really true of all the members of the CRC, all the different institutions participating. And I think it's been very helpful for me. There have been times when, for example, I couldn't get the funding for them, but they've still hung in there with me because they're all very excited about this goal, and they are very dedicated, each and every one of these institutions and various investigators. So you see Conti Rye, Michael Keating, Donna Newberg, here's Neil Kay, Bill Plunkett, Jennifer Brown, John Riven in the background, and also John Reed. And there's Bill Wigger peeking out from the side. So anyway, I think this is very important to just end on that note. And I just wanted to thank everyone. Thank you for coming here, and thank you for your participation, because I think it's your interest that's driving the process here. And I think you can't forget that. I don't think we're home yet in solving the problems of this leukemia. I think we need to push on and develop new therapies and have more options for people so we can finally say that CLL is a curable disease, and we go on with our lives without having to worry about continual HIT medicines or what have you, and worrying about the other shoe to drop. I think that's an achievable goal. So I want to thank every one of you for being a participant in that, because I think it's with your help that we can actually get that to that goal. And I also want to thank all the patients participating in all the clinical trials. I compare them to our modern day astronauts, because if you think about going into the orbit or outer space, you have to have courage, fortitude, you have to be prepared. And I think the courage that patients demonstrate to me is the same courage I see in our astronauts going to new territories like Star Trek's new worlds that have never been seen before. And it's through their efforts that we've been able to uncover better ways to treat this disease. I wish to thank them. I wish to thank in particular Laura, who's, I don't know where she is. She's also a patient advocate that is working very hard with us, and I thank her very much for her help. And also I'd like to say thank you too to Andrew Greaves here, who helped us organize this symposium and made things seamless. And so I want to thank him as well. So thank you very much for your attention. I'd like to then move on to